Hi everyone, good evening. So this is going to be the continuation of the notes in Biochem. This is going to be fatty acids part 1. This is going to be like beta oxidation of fatty acids. And then it's just going to be the notes that the professor highlighted. So out of all the slides, these are the ones that she actually specifically highlighted for us to remember for the exam. So that's why I have here. We're going to go through fatty acids. I don't know if I'm going to continue making these separate videos for the reactions themselves, since I cover them in these notes already. You know, like I already wrote all of them out and stuff. And from the first test, there wasn't really anything about the pathways. Like, you know, memorize step 5 of the pathway note. There wasn't really anything like that. So I might just do the you know, like the lecture notes that she gave us, and then kind of trimming down to where she actually wanted us to focus. All right, with that being said, let's get started. Fatty acids, these are going to be 1/3 of the energy from the dietary triglycerols, triacylglycerols. So 1/3 of your energy will come from the dietary triacylglycerols. 80% of the energy needs of the heart and the liver are going to come from oxidating fatty acids. Going to go back here. So 80% of the needs of the heart and liver are from oxidating fatty acids. Animals that are hibernating, they're going to also use fats too. So it's still as important as carbohydrates are in using energy. Okay, fuel storage of fats versus polysaccharides. So why are fatty acids and fats in general good for storage? Fatty acids will carry more energy per carbon. They are more reduced than polysaccharides. Remember, if you look at the molecule, you have a ton of carbons and a ton of hydrogens, and that's a lot of reduction. So they're going to carry more energy per carbon. They're going to also carry less water because of their nonpolar structure and just the nonpolar nature of the molecules themselves. Fatty acids, as well as just fats in general, are going to be good for long-term storage and slow delivery. The opposite is true for polysaccharides. Polysaccharides are going to be short-term storage and fast delivery. We have some other terms here. Apolipoproteins are going to be the carrier protein without the lipid. And then you have lipoproteins, which are the carriers plus the lipids that they carry. And then there's just a few types. Chylomicron, VLDL, and then HDL was the one that was highlighted in the notes that she wanted us to remember. HDL, it transports lipids to the liver, and HDL is also known as good cholesterol. All right, let's talk about the breakdown of triacylglycerol. So triacylglycerol, it stores fatty acids. Glycerol is made from the TAGs, or the triacylglycerols, and this is going to be due to the lipase. Okay, so you have this reaction here. I forgot to write the enzymes down. They weren't highlighted, but I'm going to pull those up right now as we speak. So basically you have this first step. You're going to make the glycerol 3-phosphate, and then you're going to go and then arrive to your DAP. Remember, DAP was a molecule that we found in glycolysis, and you're going to be using a dehydrogenase to get to there. Let me just scroll back to where we are. And next, the last step here, the breakdown of triacylglycerol, will be taking our triose phosphate isomerase, that enzyme again from glycolysis. The DAP becomes GAP. There's kind of the process right there. Yeah, I don't think the enzymes were highlighted. There is a dehydrogenase and then triose phosphate isomerase. Up here, the glycerol to glycerol 3-phosphate, you just use a kinase, but only the names like dihydroxyacetone phosphate and then GAP were highlighted, as well as glycerol. So glycerol becomes DAP and then becomes GAP. All right, now we get to the main point here, beta oxidation of long-chain fatty acids. So the electrons are going to be removed from the fatty acids, and then they're going to go through the electron transport chain for ATP synthesis. Acetyl-CoA will be oxidized to CO2 in the citric acid cycle from the beta oxidation. It's called beta oxidation because you are working with the beta carbon. If you're going to number the carbons in our fatty acid structure, which is like a carboxylic acid, the one I have on screen has 7 carbons. So we're going to carbon 1, that's the one that's double bound to the oxygen as well as the OH, and then carbon 2 is right next to it. Carbon 3 is our beta carbon. So all of the beta oxidation steps will be happening at that third carbon, aka the beta carbon. You're going to need to activate the fatty acid using CoAsh. So that's the co-substrate from the PDH complex part when we were going over the citric acid cycle. Well, CoAsh is also going to play a role in this too. Small fatty acids which have less than 12 carbons, you're going to have free diffusion into the mitochondria. However, with fatty acids that have 14 carbons or more, you're going to need to use the carnitine shuttle. So the carnitine shuttle will again be the way you transport these long-chain fatty acids through the mitochondrial membrane. You need the activation to a fatty acyl-CoA. So you need to make this fatty acyl-CoA. And therefore you need CoAsh in this reaction. You also need ATP because fatty acyl-CoA synthetase is a synthetase, and that requires ATP. So what will happen is you have this carnitine acyltransferase 1, and that's going to be from the cytosol to the mitochondria. And then you also have the carnitine acyltransferase 2, which is going to be the, it looks like the matrix and then into the inner membrane space, if I'm reading the chart correctly, and then cytosol too. So there's just the carnitine acyltransferase there. Yeah. Okay. Let's talk about that fatty acyl-CoA molecule now. This is the activated fatty acid. Again, you're going to take the fatty acid plus the CoAsh plus ATP. You're going to use fatty acyl-CoA synthetase. You're going to make fatty acyl-CoA, and then also AMP and PP. This is from the 2 equivalents of ATP cut. You're not using 2 ATPs, however, you're going to hydrolyze 2 phosphate bonds, so that's why it's 2 equivalents when you're working with fatty acyl-CoA synthetase. So fatty acyl-CoA has a thioester. Again, we talked about the enzyme, the synthetase that it needs. We also talked about CoAsh and ATP. It hydrolyzes 2 of those ATP bonds. All right, so here are the actual steps to beta oxidation of saturated fatty acids. You have this first step here. As an example, we're going to be using palmitoyl-CoA, which is a 16-carbon fatty acid. And the first step goes like this. You're going to have this acyl-CoA dehydrogenase as the enzyme. You have a tightly bound FAD molecule on that enzyme, the dehydrogenase, in step 1. Then what happens here, you're going to remove 2 hydrogens, and you're going to form this carbon-carbon double bond that's trans. This is known as the, in this specific instance, this is known as the enoyl-CoA. And then, of course, you're going to be making our FADH2 here, because dehydrogenase, the hydrogen is going to go in the FAD. All right, step 2. So remember, we had that enoyl-CoA, or just this double bond, the carbon-carbon double bond. This is going to be between the alpha carbon and our beta carbon. Remember, beta oxidation, it's going to be involving that beta carbon. So the carbon-carbon double bond molecule will be worked on by this hydratase. This is the enoyl-CoA hydratase. And hydratase, you're going to need water. You're going to add water to the double bond of this trans enoyl-CoA. And when you do that, you form this beta hydroxyacyl-CoA. This is the product of that, the beta hydroxyacyl-CoA. This is an alcohol. That's why I have just to an alcohol. So we went from this trans double bond carbon-carbon, we added water to it, and now it's an alcohol via the hydratase. This is very similar to the fumarase reaction in the citric acid cycle. And let me see if we need anything else here. No. Okay, let's go to step 3 then. Step 3 is another dehydration. We're going to be taking that hydroxyacyl-CoA that we made, which is an alcohol. We're going to be using NAD as well as this hydroxyacyl-CoA dehydrogenase. So we're going to use another dehydrogenase here. We're going to have NAD, and it's going to be this ketone. The enzyme is stereospecific for the L stereoisomer of this alcohol, this beta hydroxy acid, acyl-CoA. So we need the L version of this, the L stereoisomer. And then again, dehydrogenase, we get this ketone known as a ketoacyl-CoA. This is the product of step 3. So we went from an alcohol to a ketone. And this is similar to the malate dehydrogenase reaction of the citric acid cycle. It's very similar to the malate dehydrogenase enzyme. Okay, we also get NADH here too. All right, step 4, we're at the end of beta oxidation. Okay, so acyl-CoA acetyltransferase, also known as the thiolase, that's going to be our enzyme here. We're going to take our ketoacyl-CoA, which we've made in step 3, this ketone, and we're going to also require CoAsh in this reaction with the thiolase enzyme, and we're going to make acetyl-CoA, and we're also going to have this fatty acyl-CoA left over, but it just has -2 carbons from the start, so this would be a 14-carbon fatty acyl-CoA. Remember, we started with a 16-carbon one. After one go of beta oxidation, we make acetyl-CoA plus a fatty acyl-CoA that's 2 carbons less than what we started with. So therefore, we could actually go through this cycle multiple times. Acetyl-CoA here will go through the Krebs cycle. And then, just to clarify, let's see. Okay, so 16 carbon will have 1, 2, 3, 4, 5, 6. We'll have six removals basically, and then it'll just, it's a cycle basically. Okay, so let's talk about the overall reaction. Each pass of the beta oxidation will remove 1 molecule of acetyl-CoA. We're going to have 2 pairs of electrons and 4 protons. So that means 1 acetyl-CoA, 1 FADH2, 1 NADH, and 1 hydrogen per pass. Okay, so we also have this formula here. So the formula is like a quick and easy way of finding the amount of all of these products. So let's say we have a fatty acyl-CoA of X amount of carbons. The products will equal X divided by 2 for the acetyl-CoA, so half the number of X. And then for FADH2 and NADH, that's going to be taking half of whatever X is, and then you're going to subtract 1 from that. So in our example, if we had the 16 carbons, that would lead to 8 acetyl-CoA. And then for FADH2, that would be half of 8 minus 1, so that would be 7. And for NADH, same process. It would be half of the 16 carbons, which is 8, and then U minus 1, so 7 NADH, and then 7 hydrogens as well. Okay, so now we could then use the numbers here, the formulas for how much ATP is generated ultimately from the carriers. So FADH2 will actually equal 1.5 ATP. So you take the number of FADH2 and you multiply it by 1.5 to figure out how much ATP you've made. NADH would be 2.5 ATP. So in that regard, let's say we took our palmitoyl-CoA and we want to know the amount of ATP after the beta oxidation and then also the citric acid cycle of the acetyl-CoA that we make. So again, using our formula, we would get 8 acetyl-CoA and 7-FADH2 and 7-NADH for palmitoyl-CoA after beta oxidation has completed. And then we would go into citric acid cycle with those 8 acetyl-CoA. Each turn of the citric acid cycle per acetyl-CoA will be 3 NADH, 1 ATP, and 1 FADH2. So we would take the 3 NADH, multiply it by 8 to get to 24 NADH. We would take the 8, multiply it by the 1 ATP, and then that's 8 ATP. We would take the 8, multiply it by the FADH2, 1 FADH2, so that would also be 1 FADH2. Okay, after that, we would find that the 7 FADH2, we would add it to the 8 FADH2 from the citric acid cycle to get to 15 FADH2. Now we take the 15 and multiply it by 1.5 to get to 22.5. We take the 7 NADH from beta oxidation of palmitoyl-CoA, which is a 16-carbon molecule. Then we would add this to the 24 NADH that would come from the citric acid cycle of the 8 acetyl-CoA. That would get us to 31 NADH. Now we take the 31 NADH and multiply it by 2.5 to get to 77.5. That would get us to 100 ATP, but we still need to add the 8 from the citric acid cycles, 8 acetyl-CoA. So 8 plus 100 is 108 ATP for palmitoyl-CoA, 16-carbon fatty acid. Alright, so now we could also talk about the monounsaturated fatty acids. So these ones are going to be requiring a delta 3, delta 2, sorry about that, enoyl-CoA isomerase. So we're going to need to use this enoyl-CoA isomerase with delta 3, delta 2. And you're going to take the cis-enoyl-CoA, delta 3, to a trans molecule of delta 2 enoyl-CoA. Oxidation of a polyunsaturated fatty acid, you're going to also need this enoyl-CoA isomerase, this in addition to the 2,4-dienyl-CoA reductase. So you need both the isomerase and the reductase for a polyunsaturated fatty acid. Right, let's talk about odd number fatty acids now too. So first we have to talk about this molecule, propionate, which is a 3-carbon compound basically formed by cattle from the carbohydrate fermentation. However, when odd number fatty acids are beta oxidized, they make acetyl-CoA as well as this molecule here, propionyl-CoA, which is a 3-carbon with a CoA at the end. Many steps will be involved, but then ultimately this will become succinyl-CoA. Succinyl-CoA is, of course, involved in the citric acid cycle. We could also have the peroxisomes carry out beta oxidation. So they're going to be more active on long and branch fatty acids. So the peroxisomes like long and branch fatty acids. It's the same 4 steps that we went over. We have the dehydrogenase, and that's going to take the fatty acyl-CoA to a carbon-carbon double bond. Hydratase will turn that carbon-carbon double bond into an alcohol. I'm going to add water in that step. And then remember dehydrogenase as step 3, that's going to turn that alcohol into a ketone. And then thiolase is our final step that's going to split it into acetyl-CoA and then a fatty acyl-CoA that's negative 2 carbons, or 2 carbons less, I should say, from your starting fatty acyl-CoA. Okay, we also have the ketone body, or ketone bodies. And when oxaloacetate is low, acetyl-CoA is converted into the ketone bodies. This will free up CoAsh for more beta oxidation, and some of these ketone bodies include acetone, which is exhaled. We also have acetoacetate, as well as DB hydroxybutyrate. Those are just 3 types. Okay, so the liver is a ketone body source. The conditions that cause gluconeogenesis, as well as untreated diabetes and severely reduced food intake, will deplete oxaloacetate and slow the citric acid cycle. The acetoacetate and that DB hydroxybutyrate molecules, those ketone bodies, are going to be transported to the extra hepatic tissues to be converted into acetyl-CoA. All right, that's going to be all on beta oxidation for now. Again, remember, beta oxidation, 4 steps. After we activate our fatty acid with CoAsh and ATP, we're going to first do that with fatty acyl-CoA synthetase, and that's going to be 2 equivalents. Let me make sure there isn't any, like regulation. Let me go to the very end here. I forgot to add the regulation in. Okay, synthesis, oxidation. So regulation of fatty acid oxidation. So we're going to stop the carnitine shuttle by this malonyl-CoA, which we'll talk about in the next video when we're talking about synthesis. But the first intermediate of cytosolic fatty acid synthesis also blocks the entry of the fatty acids into mitochondria. It stops the carnitine shuttle. This prevents the simultaneous synthesis and degradation of fatty acids. Then I think the rest of them are, the rest of the regulation portion, I guess we'll just cover it in the next video about the synthesis of fatty acids. All right, thanks so much for tuning in. Please do something nice for someone and take care.